Okay, guys, today's topic for the seminar is the muscle tissue. And before we begin, I wanted to ask you, what do you think the function of a muscle is? A basic function.
Movement.
Exactly. So the muscle tissue basically helps in movements of our entire body. And the way it does it is with constant contraction and relaxation of the muscle fibers. So the muscle fiber is basically the smallest unit of a muscle, and it is made up of myofilaments. And these myofilaments are of two types, actin and myosin, and they are arranged in a specific way to form a muscle fiber. Now, these two terms, which are written in this pink box, they are going to be used often throughout this seminar. So, the sarcoplasm is just another name for a cytoplasm, but it is the cytoplasm of a muscle. That's why it is called as sarcoplasm. And the same thing applies for sarcoplasmic reticulum. It is just another name for the endoplasmic reticulum of the muscle. So let's talk about the skeletal muscle coverings. There are three main coverings we are going to deal with over here. There is the endomysium, the perimysium, and the epimysium. So the smallest thing in a muscle is going to be the muscle fiber, as you can see in this diagram over here. These small circular things are the muscle fibers, and the covering for that is going to be called as the endomysium. As since it's the innermost covering, that's why it's called the endomysium, and it is made up of the loose connective tissue. The purpose of this loose connective tissue is basically to supply it with blood, and it also has lymphatic function, and there's also the nervous stimuli which is sent through this loose connective tissue. Now, multiple of these muscle fibers, they come together and they form something called as the primary muscle bundle, which is again enwrapped with this loose connective tissue, and this thing is called as the perimysium. In large muscles, multiple of these primary bundles can also join to form a secondary bundle, which is again going to be covered by the perimysium. The when majority of these primary and secondary muscle bundles, they combine together to form a really big structure, then that is going to be covered by something called as the epimysium. That's going to be the outermost covering, and the thing that joins the epimysium to the bones or to the surrounding muscles is going to be called as a fascia. This fascia is made up of dense connective tissue. That's the difference between the fascia and all the other coverings. All the other coverings are necessarily made by the loose connective tissue except for the fascia. Now, when you look at them in your slides in your histology class, you're going to be able to see something like this, like these kind of pink structures, and in light microscopy. So you can view it in two perspectives. There's going to be a longitudinal perspective and a cross section. So the longitudinal perspective basically means like if you hold something like this and you cut it like that through it, and whatever you're going to be able to see in that section is going to be called as a longitudinal section. But if you're holding something like this and you cut it midway, then whatever you're going to be able to see is called as a cross section. The important thing to note over here is that the nuclei in both of these sections is going to be visible just beneath the sarcolemma. As you can see here, like they are flattened and they are pressed towards the end, even over here. Now, satellite cells, does anyone have any idea what their function is going to be like? All right, okay then. Basically, they lie on the surface of every single muscle fiber, and they're really tiny cells, and their basic function is regeneration. That's why they're considered as the adult stem cells, and they offer very limited regeneration, and that is also possible only if the basal lamina is not damaged in any way. If it's not damaged, then they kind of become activated to repair like really small damages. And the important thing to note here is that they are mostly act on skeletal muscles. Now, the myofilaments I was talking about earlier, so they are going to be the basic elements of contraction, and they do not shorten. The only thing they do is that they slide on each other, which causes contraction, and when they like do not slide on each other and they move away from each other, that relaxes the muscle. There's two types of myofilaments. There are thin myofilaments and the thick myofilaments. The thin myofilaments here are the actin, this blue balls over here, they're called the actin, and this thread-like structure is called the tropomyosin, and these three molecules over here, they are the troponin. So the actin molecules are made up of polymerization of G-actin, which results in the formation of an F-actin. And the tropomyosin just basically holds all of the structure together. And the troponins, as I've mentioned, there's three molecules, and each three of them have their own individual function. They are divided into three parts, like you can say TNC, TNI, and TNT. The T1 stands for tropomyosin. So the T troponin binds to the tropomyosin. The C1 binds to the calcium ions, which are released by the sarcoplasmic reticulum during the mechanism of contraction, and the I1 binds to the actin itself. The thick myofilament is called as the myosin, and it is really very thick. It is twice thicker than the thin filament, and it is joined at the structure called as the M line. The important thing to note over here is that their heads are pointed away from the M line, and that is because if the head is pointed away from the M line, then it makes a contraction really easy. Like if they will just like move this way, so the contraction is made much more easier that way. So the sarcomere is the contractile unit of a muscle, and it is between two Z lines. So these Z lines are the structure to which the actin is going to be attached to. This is whatever I'm saying right now is like the most basics, and you have to know this if you want to understand muscle contraction and everything else. And it's also very important for your micros because they ask you the function of this or what attaches to what. So the Z line is going to be this line, zigzag line over here, to which all the actins attach to. The M line is going to be the structure over here to which the myosins will attach to. Now, the I band is going to be the band, which is the light band, and only where the actin is present, that part is going to be called the I band. The entire part from where the myosin is present is called as the A band, and the A band has two zones. As you can see here, this dark zone over here, that is where there's both actin and myosin present. So this is going to be a main contractile part of the muscle fiber. But over here, you can see only the myosin is present, and this part is called as the H zone. Does anyone have any questions about what I just explained to you? Okay, I'll move on. So, like I mentioned earlier, the sarcoplasmic reticulum is called as the endoplasmic reticulum of the myofibrils, and it surrounds each and every single myofibril. The thing is that there's two parts over here. There's longitudinally oriented part and a transversely oriented part. The important part over here is only going to be a transversely oriented part, which is called as a terminal cisternae. You can see this blue structure here and this blue structure here. These all of these structures are called as the terminal cisternae, and this orange thing which is coming out of it, which is protruding out, is called as a T tubule. Two of these terminal cisternae and one of these T tubule makes up a structure called as a triad, and this triad is an important word to remember because this is a characteristic only for the skeletal muscle. The function of these T tubules is like it transmits all the nervous impulses, which helps in depolarization of the sarcolemma, and it results basically in uniform contraction. The function of this SR is like it helps in the muscle contraction. And there's three basic steps about muscle contraction which you need to know is that when the muscle is relaxed, the calcium ions they accumulate in the cisternaes, and when the it's about to start contracting, the sarcolemma depolarizes, and all the calcium ions they come out from the sarcolemma, and they go around the myofibrils, which enables contraction. And the contraction process ends when all of these calcium ions they return back to their cisternae. That is when like the muscle goes back to its relaxed state. Now, the motor end plate is basically another mechanism of, you can say, contraction. And the contraction basically begins when acetylcholine is released from the motor end plate. And the purpose of this acetylcholine is to increase the permeability of the sarcolemma for sodium ions, because the permeability of sarcolemma is increased for sodium ions. The sodium ions will go in, and this causes the calcium ions to go out into the sarcoplasma, which again initiates contraction. And this contraction is basically done when the actin filaments they slide on top of the myosin filaments. And like I said earlier, the contraction stops when the calcium ions they again return back to their SR. Now, Varun will continue from here. Does anyone have any questions
Um,
regarding whatever I said so far?
yes, I do.
All right, go ahead.
There was one slide where you were explaining about some structure that was blue color and an orange color. I did not hear their names properly.
Oh, okay, one second. Let me go back to sharing my screen. So, sarcoplasmic reticulum. Okay, so the only thing you need to know about this slide is that there is a transversely oriented structure which is just called as the terminal cistern A. You can see this blue part over here, right? So it is transversely oriented because it's like flat, like it's horizontal. So this structure is going to be called as a terminal cistern A. It's just a thing you have to remember. And this orange structure is called as a T tubule. This is also a thing you have to remember. These are just the structures with their specific functions. And you can see here one blue thing, and then there is one more orange thing in the middle, and there's one more blue thing. So these three things they combine together to form one unit, which is called as a triad. This is just a bunch of information. It does not really correspond to any particular function. It's just that these are basic facts which you just have to memorize, kind of. The only thing you need to remember here is the function of a T tubule. The T tubule just is involved in spreading of the nerve impulses, which help in uniform contraction. That's the only function you need to know here. Apart from that, it's just there's one terminal cistern, another terminal cistern. Between them, there's a T tubule, and these three things just form a triad. Is that okay?
Yes, okay, thank you so much.
No problem.
Guys, you can see my screen.
Yes, it's visible.
Yeah. All right. So before I begin, Ria had mentioned about A band. So A band is anisotropic, and that's why it cannot like let light pass through. That's why we call it A band. A band anisotropic. So that's just something I wanted to add on. And now we'll begin from the skeletal muscle contraction mechanism. So here in the PPT, I've added a YouTube video. You guys can watch it later on your own time for like better understanding. It will help you, visualize this information. And yeah, let's begin. So skeletal muscle contraction. ATP binding and hydrolysis. At rest, ATP binds to myosin heads, but it breaks down slowly. And this ATP is essential for the release of energy for muscle contraction. So basically, as Ria had mentioned, there's two components. We had the myosin and actin. So initially at the resting phase, ATP just binds to the myosin, but there's nothing happening. It's just breaking down slowly, but there's no contraction or relaxation happening. After that, role of actin and myosin. So normally, as I said, myosin cannot attach to the actin because a troponin tropomyosin complex blocks the binding sites on the actin. So basically, the troponin tropomyosin complex will act as a barrier for the myosin, so it cannot attach to the actin. So in order to go over this barrier, we have calcium ions which release from the sarcoplasmic reticulum, and these change the shape of troponin and free the binding sites, allowing the myosin to attach to the actin. After that, what happens is that, the myosin particles, they have the ATP cap on top. As we said, at the rest, they had ATP attached to them. This ATP cap, it gets broken down into ADP and phosphate, and the energy released is allowed for the contraction. So here on this image, you can see initially we have the myosin binding sites covered by the troponin here. And then when the calcium is released from the sarcoplasmic reticulum, the myosin sites are exposed, allowing them to attach. And then what happens is that after the contraction happens, yeah, we need another ATP molecule to allow the actin, to allow the myosin to detach from the actin and allow another cycle to begin. what will happen is if there's no ATP here, this will stay in the same contracted position and it will result in rigor mortis, which means that muscle stiffness. This usually happens after a person's death when there's no muscle contraction happening. And contraction cycle continuation, the cycle of binding and contraction and detachment continues until the calcium is pumped back into the sarcoplasmic reticulum and it requires more ATP for its relaxation. So till the person is alive, ATP is made and calcium ions are moving into and out of the sarcoplasmic reticulum. This contraction mechanism will happen, otherwise it will not happen. So and not just to summarize this entire mechanism, I'll say it in more easier words than it was on the slide. So the entire mechanism of the contraction is, like a tug of war game in which we have the myosin and the actin filaments. And myosin has this energy particle called ATP, and actin is just there, which has a blocker particle on it called the troponin tropomyosin complex. So first of all, the calcium ions will come on and they will remove this blocker, the troponin tropomyosin complex, and then the myosin will basically use the ATP, which is like the energy snack, and it will pull the actin filament to the side. Now we have to release this force, otherwise this will remain in the same position. So we need another ATP particle to bind to the myosin to allow the detachment to take place. And after that, during the relaxation, calcium is pumped back again into the sarcoplasmic reticulum, and ATP is needed for another calcium reuptake. So, guys, does anyone have any questions about this? No? So, we'll move on. Now, we talk about cardiac muscle, or it's also called myocardium. So, the structure of this is that we have a morphological unit which is called cardiomyocyte, and these cells are organized into chains and they are connected by intercalated discs. Now, it is important to remember that these intercalated discs, they're only seen on the slide in a longitudinal section, not in a cross section. I'll show you that in the next slide. So what happens is that these intercalated discs, they give the myocardium a network-like structure, which allows for, strong coordinated interactions. So in your microtest, you can have a question like, select the features of the myocardium, and the options can be intercalated disc, net-like structure, blah, blah, blah. So that's the format of the questions in the microtest for this topic. And then function would be, it enables the rhythmic contraction of both the atria and ventricles to allow pumping of oxygen throughout the body. And then vascularization, it has the highest vascular density inside the body. Why? Because again, we need rhythmic contraction of the heart to supply oxygen throughout the body. Now, unlike skeletal muscle, which had, the epimysium, perimysium, and endomysium, the cardiac muscle, it only has endomysium. As you can see, these white spaces, this represents the endomysium, which is just nothing but, the loose connective tissue, capillaries, and nerves. So this is the longitudinal section where you can see these cardiomyocytes like this, and these white spaces is the one which represents endomysium, and here also. And these small things, these large diameters, these are cardiomyocytes, cells with large diameter, and the ones which are small here are capillaries. And then when we talk about the innervation of the myocardium, it is innervated by the autonomic nervous system. So the autonomic nervous system has two parts. First one is it's also called the fight or flight. So the first one is the parasympathetic system, which will slow down the heart, and we have the sympathetic system, which will increase it up. And now, when we talk about the regeneration of the cardiac muscles is not possible. So, for example, if they will get damaged, that would be a permanent damage and they will not be regenerated again. About the contraction mechanism, it's just the same as the one in skeletal muscle, but it is more adapted for continuous contraction rather than whenever it is needed. So on this diagram, you can see again, this is a longitudinal section. These lines, they represent the intercalated discs. And then we have these P's represents the cytoplasmic processes, which basically connect, the network of cardiomyocytes. So again, I just want to ask which one, I mentioned this, but which one is the longitudinal section and which one is the cross section from these images?
The first one is the longitudinal one.
Yeah, and the other one is the cross section. So just to summarize all the differences I said, Longitudinal section, you can see cardiomyocytes are arranged in a columnar arrangement, and they are connected by their cytoplasmic processes. And I said abundant capillaries, it is highly vascularized and longitudinally arranged striated myofibrils as well as intercalated discs. Everything is labeled here. You can see these are the intercalated discs. This is again the endomysium with capillaries, nerves, and this is the nucleus of the cardiomyocytes. And the cross section, like I said before, large diameter would represent the cardiomyocytes here, and this center is the nucleus, and smaller diameter would represent the cytoplasmic processes. Does anyone have any questions about the longitudinal and the cross section? All right, next. So again, just the characteristic, the question can be select characteristics, all the characteristics or the statements which are true about cardiac muscle, network-like structure, numerous capillaries, numerous mitochondria, around 20 times more than skeletal muscles. And again, the intercalated discs are seen in the longitudinal section, and the cell is covered by the basal lamina with few reticular fibers and collagen fibers, which form the basement membrane. And we studied this for the first micro test, so I hope you guys remember some of it. And then we have the organelles and the sarcoplasm. They're rich in organelles like glycogen, Golgi apparatus, as well as lipofuscin. Now, sarcoplasmic reticulum in the cardiac muscle versus the skeletal muscle. So as Ria had explained, sarcoplasmic reticulum, it's nothing but like, a storage center for calcium ions. So in cardiac muscle, it is less developed compared to skeletal muscles. So it has less amount of calcium present in it compared to the skeletal muscles. So that's why it has partially developed L cisternae as well as terminal cisternae. And then when we talk about the T-tubules, these are wider and more numerous than in cardiac muscles. So T-tubules, these are, special pathways, and they are longer compared to, in the skeletal muscle, because we need, the continuous contraction of the heart muscle to provide oxygen throughout the body. That's why they are wider and more numerous than the skeletal muscle. And now the T-tubules, they are only attached to one terminal cistern, which forms a diad compared to a triad in the skeletal muscle, as again Ria explained before. Now, in both cases, the sarcoplasmic reticulum surrounds a single myofibril. So why does it surround a single myofibril? This is because it has to make sure that the calcium ions, they're delivered exactly where they're needed. So that was the difference between the sarcoplasmic reticulum in both. And now we have the intercalated discs. Intercalated discs, as I said, they're seen in longitudinal sections. They connect the cardiomyocytes. So they have two parts. We have the transverse portion and the lateral portion. So the transverse portion is made up of fascia adherens and desmosomes. Now I have highlighted macular adherens as well as nexuses because these are, the Latin terms for these words, and they can ask in the questions like what components are part of the transverse portion, and they'll not say desmosomes, they'll say macular adherence. So you just have to know that these are the same thing, and it's just the Latin term of it. So when we talk about the transverse portion, it includes fascia adherens, which anchors the actin filaments, and desmosomes or macular adherens, which bind the cell tightly. So it's just, like a gripping portion of the intercalated discs. And when we talk about the lateral portion, it contains gap junctions or nexuses, which allows the ions to pass between the cells and facilitate the spread of the contraction signal. So again, you can clearly see this on the image. This is the transverse portion. This shows the zonular adherence, and this shows the macular adherence, which is between the cardiomyocytes. And then this is the lateral portion. Now, when you talk about the Purkinje cells and the fibers, so Purkinje cells, they originate from the cardiomyocytes, and they are critical for regulating and propagating the heart's rhythmic contractions. So where they are found underneath the endocardium, but above the myocardium in the space between both of these layers. So I'll explain that later. And then, yeah, here. So you can see here we have the endocardium here, which is the layer above the Purkinje cells, and then this is the myocardium. This is the slide of the heart, which you will study for the next topic as well. So it's better to know that now as well. So this is endocardium, which is the topmost layer or the jagged layer, which we call it. And then we have the Purkinje cell layer, and then we have the myocardium, which is like the working layer. Then structural features compared to cardiomyocyte. They are large, Purkinje cells are large with a central dark nucleus, and they're binucleated. And it is important to remember that they have few myofibrils compared to, the cardiac muscle. And then These cells, they rely on anaerobic respiration due to their low number of mitochondria and accumulation of glycogen granules. So they appear lighter on the slide. And then when we talk about the sarcoplasmic reticulum, it is reduced and the T tubules are absent. And the intercalated disc, they only have, desmosomes, which was the part of the transverse portion, and they only have few gap junctions or nexuses present. Here we can see this on the slide better. So when we compare the Purkinje cells with the cardiomyocyte, here you can see the cardiomyocyte and the Purkinje cells. So you can obviously that the Purkinje cells, they are larger and they are lighter. Why? Because they have fewer mitochondria and they have abundant glycogen granules. And you can see some of them are binucleated as well. Here you can see two nucleus and cardiomyocytes are not. So that was the difference between the Purkinje cells and the cardiomyocytes. Does anyone have any questions about this? No, so Katrina, you can.
Okay. Oh, sorry. Give me a second. Okay. Can you guys see my screen? Hello? Hello? Can you guys see my screen?
Yeah.
Thank you. So we're going to talk about smooth muscle. This is the last part of our seminar, so I know you guys are tired, but please just hold on for a little longer. So smooth muscle, the morphological unit is the myocyte. The myocyte is a spindle-shaped cell, and it's the smallest element of smallest muscle element of smooth muscle. Smooth muscle contains myofilaments, as you guys already know, actin and myosin, and in smooth muscle, they're irregularly longitudinally arranged, and unlike skeletal, they do not form myofibrils. So you are not going to see any cross striations in smooth muscle. You also have intermediate descendant filaments, which are going to connect the dense bodies. The sacrolemma is connected with numerous caveolae. This is very important. There is no T system. There are no T tubules. You have caveolae, and they help in the regulation of calcium ions concentration in the sacroplasm, which you guys know are necessary for the process of contraction. So the important things to take: smooth muscle, myocyte is the morphological unit. They do have myofilaments, but these myofilaments do not form any myofibrils, so you guys will not see any cross striations, and there are no T tubules or there's no T system. Instead, you have caveolae. And this last part is very important. If you guys get any sort of slide that has muscle, they will ask you about the difference between smooth muscle and other muscles. And it's very important to mention that they do not have a T system. Okay, in the light microscopy, as Varun was mentioning, when we look at muscles, we can look at them through a longitudinal section and a cross section. In a longitudinal section, you guys can see spindle-shaped small cells, and centrally you're going to see one dark elongated nucleus, and as you guys can see, you will not see any sort of myofibrils. Cross section, you're going to see a great range of diameter of cells, and larger cells will contain centrally located nucleus, which is what you guys can see with my mouse. These are the centrally located nucleus in the larger cells, but as you guys can see, not all cells will have a centrally located nucleus. The activity of smooth muscle is weak, it's slow, and you're going to have involuntary contraction. So myocytes, these are some important components. You're going to have the endomysium, which is the connective tissue that is going to surround each muscle cell. And the endomysium is important. It's already been mentioned, but I'll mention it again. It can conduct nutrients via the vessels. It helps with innervation and spreading of contraction. Innervation of myocytes. So you have autonomic, sympathetic and parasympathetic. The nerve axons will terminate in the endomysial connective tissue. Myocytes, they can receive nerve stimuli over the entire surface, and they can have also spontaneous activity. And the conductance of the stimuli across the gap junctions will enable them to contract synchronously or all together. There's also supporting function. Myocytes are able to produce components of extracellular matrix, and their regeneration is very limited. So now the mechanism of contraction, it's going to be a bit similar. I have an image later to help you guys better understand, but you guys know that the thin filament, myofilament is actin, the thick is myosin. Here, the actin will consist of actin and tropomyosin. Troponin is going to be substituted by calmodulin, and this is mostly going to be inserted into dense bodies. These are some of the differences that are important for you guys to know. So in smooth muscle, the thin myofilament actin is going to consist of actin and tropomyosin. The troponin is going to be substituted by calmodulin. The thick myofilament myosin is seen only in a relaxed state. And there is going to be a greater degree of smooth muscle contraction than of the skeletal muscle fibers. You have the intermediate filament, desmin, and again, I will show you guys an image later after the slide so you guys can better visualize it. But desmin is basically going to form this lattice-like network, and it's going to interconnect the dense bodies. And this helps in participation of the propagation of the contraction wave. And then you have the dense bodies. There are two types of dense bodies. You have the membranous and the cytoplasmic, and they basically correspond to like it's the smooth muscle version of a Z-line. So they are anchoring structures. And this is basically everything. So here you guys have the dense bodies, which are like the anchoring structures, and you guys see that they're being connected by the intermediate filaments. Intermediate filaments here. And you guys can see thick filaments, and here you have the thick thin filaments, actin and myosin. And this is just another image again to help you guys better visualize dense bodies, the intermediate filaments, and then here you have the thin and thick filaments. 1, 2, 3, 4. Hope that that's good enough for you guys to visualize what we see on the cell. So again, it's really similar when we talk about how the contraction works. We have the sliding filament theory. Basically, the sarcomeres are going to shorten, and that's what's going to cause the contraction. We start with the first step, which is ATP hydrolysis. The ATP is going to be hydrolyzed, and this is going to change the angle of the myosin head. And the myosin head is going to be ready to bind to the actin. Then the calcium is going to bind to troponin and expose the active binding site on the actin. So now the myosin head will be able to bind to the actin. And here we have our official cross bridge. And then we have our power stroke, which is when the sarcomere is going to shorten. So then you have the myosin kind of pulling the actin, and then which is our contraction. And then we're going to have detachment once you have another binding of an ATP molecule. This is just one of those things that you guys are just gonna have to kind of like, try your best to learn it, try to explain it to a friend. It's 4 steps. It sounds complicated, but it's pretty simple. Watch as many videos as you can. And once you guys learn this, it's going to help you so much, because this is one of those things that you do not want to learn this for the first time before your final. So again, this is just, I wrote down the sliding mechanism, but I think the image before was just much better. You guys know there's going to be an influx of calciums. They help initiate the contraction. But remember that when we're talking about a smooth muscle, they're going to bind to calmodulin, not troponin. The ATP, just some important things to know, that the ATP breakdown is going to be relatively slow. So the contraction of smooth muscle is slower, and that's why you have like less fatigue. The sliding, as you guys know, is going to result in the contraction because the cells are going to become shorter, and smooth muscle, in comparison to the skeletal one, is able to keep a strong force of contraction. Don't confuse slow and strong. What I mean to say is it's slow and strong, but it is strong. And this is with very little ATP utilization. So there are some other types of cells that you guys that have myofilaments and have contractile abilities. These are the myoepithelial cells, myofibroblasts, and pericytes. So they contain like actin with calmodulin, myosin, and desmin. So first we can talk about myoepithelial cells. Myoepithelial cells, they have one oval centrally located nucleus, and they have the myofilaments. Myoepithelial cells are mostly found in glands. When we did our seminar about glands, I don't remember if I mentioned it, but I did show you guys a picture, which is similar to this one in the corner. So these are the myoepithelial cells. And basically, they help with contraction. They help drain the secretory products. You have them in the salivary glands, skin glands. They are not in the sebaceous glands. And can anyone, I understand if you guys don't know, but if anyone knows, do you know why they are not in the sebaceous gland? Basically, we just said that the reason why myoepithelial cells are found in all these glands, in the mammary gland and whatever, is because they help with contraction, right? They need to contract, so then you have secretion of the product that the glands are going to make. Well, in the sebaceous gland, it actually has a muscle. I forgot the name of the muscle. And that muscle kind of does that job, and that's why you won't see the myoepithelial cells there. Function, again, responsibility for drainage of the secretory products, and innervation is autonomic or involuntary. Then you have the myofibroblasts. These are connective tissue cells, and basically they secrete collagen. They contain myofilaments. They do not possess a basal lamina, and they are present in regions that have loose connective tissue, like the alveolar septa of the lung. Basically, myofibroblasts, just know, guys, that they are important for healing because they secrete collagen. Imagine that you guys cut yourself or just get hurt, you scrape your skin. The myofibroblasts are going to start secreting, they're going to start proliferating and secreting collagen to help with the healing process. And note that in some pathological conditions, and by pathological I mean when some people might have some disease that's going to cause extreme like proliferation and secretion of collagen, and this can cause like cirrhosis, liver cirrhosis, lung cirrhosis. And then we have parasites. Parasites are, again, these are all contractile cells, myoepithelial, myofibroblasts, and pericytes. They're all contractile cells. And these contractile cells, they surround capillaries and venules. And after you have tissue injury, they will proliferate and they're going to substitute undifferentiated mesenchymal cells. So they can differentiate into myoblasts and fibroblasts. This is not as important as myofibroblasts. So you guys, just like study it very, very basically. You probably will not get asked about this one, but myoepithelial and myofibroblasts 100%. And now this last image is an image that I got from Yo's lecture. And this just shows like the difference of all the types of muscles that were discussed in today's seminar. I just going to quickly go over it, but again, it's in the lecture. So we know that skeletal muscle, we have the muscle fibers, cells, cardiomyocytes for myocardium, and smooth muscle, the cells are myocytes. And in a longitudinal section, we know we're going to see for skeletal muscle, large muscle fibers that are parallel. We're going to see a lot of flat nuclei beneath the sacral lemma. And you're going to see striated myofibrils throughout the whole sarcoplasm. For myocardium, in a longitudinal section, we're going to see cardiomyocytes that are interconnected by 3D cytoplasmic processes. Each cell will have one or two oval centrally located nucleus. So for skeletal, the nucleus is beneath the sacral lemma, as you guys can see here. For myocardium, the nucleus is centrally located, and you're going to have longitudinally arranged striated myofibrils, intercalated discs, and for smooth muscle, we're going to see spindle-shaped small cells, centrally located, one dark elongated nucleus, and there are no myofibrils. Just please remember that smooth muscle has no myofibrils. That's like probably one of the smartest things you guys could say if they ask you to differentiate between them. And then cross-section, large round fibers of the same diameter. For myocardium, larger structures, cardiomyocytes, the smaller ones, cytoplasmic processes. And only a small number of the larger structures will have a centrally located nucleus. And for smooth muscle in the cross section, again, there's a great range of diameters. You're going to see cells of all different sizes, and only the largest profiles will have a nucleus. I really recommend that you guys study this last image really, really well. It's going to help you guys a lot. And that's it from me.
Does anyone have any questions? All right, okay then. Everyone, thank you so much for coming. We hope to see you next week as well. Have a nice day.
Toodles.
Thank you.